Hello, welcome to today's video. My name is Edward. Our topic for today is virus isolation through cell culture. By the end of this lesson, you should be able to define virus isolation, define cell culture, understand the process of culturing viruses, and understand the importance of cell culture of viruses. To introduce us to this topic, viruses are obligate intracellular organisms. This means that viruses need a living host in order to replicate. Unlike bacteria, they are not capable of reproducing on artificial nutrient media. This is because viruses lack ribosomes, which are essential in the synthesis of proteins and energy. Clinical diagnosis of viral infections depends on the isolation and study of the virus in a safe and conducive environment. Virus isolation is defined as the process of separating and collecting the virus from the infected host or material from the host. It is an important method for the clinical diagnosis of viral infection. Virions, also known, which is another name for virus particles in liquid medium, can be separated from the host cells either by centrifugation or filtration. To better understand this, in the collection of a sample from an infected living host, that sample or living tissue is collected into a centrifugation tube, and after centrifugation, the cells release and release the virions into the supernatant, and the least cells now will settle to the bottom of the tube, forming the debris. We will collect the supernatant and pass it through filters of the pore size 0.2 micrometers in order to collect the virions into a separate collecting tube. Culture of viruses can be defined as the growth or propagation of viruses on living cells for the purpose of detection and study. The culture of viruses can be categorized into two: in vitro category, which is the first, is culturing viruses outside of a living organism, but in living cells and in an artificial environment with artificial nutrient media, such as in a culture flask or in a petri dish or a tube. An example of this is the tissue culture system, also known as the cell culture technique. The second category is the in vivo culture of viruses. This is the culture of a virus in a whole living organism, such as in a plant or an animal. An example is the embryonated egg system. The second example is the whole animal system, such as natural host, experimental animals, and transgenic animals. In order to appreciate the progress in this field of culture of viruses, we need to look at the history. In the 1900s, embryonated eggs and lab animals were used in the isolation of viruses. In 1913, the vaccinia virus was grown in culture for the first time. In the 1930s, yellow fever and smallpox viruses were grown in culture for the first time. In 1950, polio virus was isolated for the first time. In the 1960s, isolation of viral pathogens in cell cultures commenced. In the 1970s, something important happened whereby commercial development of purified reagents and cell lines began, and this helped in solving the limitation which affected the previous years. This led to, to advances in the development of cultures, the development of vaccines, and also in the study of viruses. Cell culture technique is considered the gold standard for virus isolation because it is more accurate and sensitive. A primary cell culture is freshly prepared from animal organs or tissues. Cells are extracted by mechanical scrapping or mincing to release cells, and then the enzymatic method is applied whereby trypsin and collagenase enzymes are used to break up tissue and release cells into suspension. The primary cell culture is then grown in culture flasks using liquid culture medium because this cell culture exhibits anchorage dependency. That means it needs to attach to a surface in order to grow. And this leads to contact inhibition when the cells have already covered the entire surface of the culture flask. This contact inhibition is caused by the cell-to-cell interaction after the cell population has increased. This cell-to-cell interaction will turn off the mitosis and thus turn off the growth in population. That requires subculturing that cell culture into a secondary flask, which we will now call the secondary cell culture. Continuous cell lines, on the other hand, are derived from transformed cells or tumors and can be subcultured many times. An example of this cell line is the HeLa cell line, which is a short form for Henrietta Lacks cell lines. Continuous cell lines do not have anchorage dependency and therefore are not really limited by contact inhibition. Examples of other cell lines widely used in the labs include Vero cell lines, the rhesus monkey kidney cell lines, human foreskin fibroblast cell lines, and others. There is a picture of a T25 cell culture flask with media. On the other side is a picture from a microscope showing a confluent monolayer of Vero cell lines in a cell culture flask. The liquid artificial media used in the cell culture technique consists of a substrate that supplies essential amino acids, minerals, growth factors, and gases such as oxygen and carbon dioxide. Antibiotics are introduced to prevent contamination from bacteria and fungi. Once inoculated with a virus which has been decontaminated and purified, the cell culture is incubated at specific conditions such as 37 degrees temperature, 5% humidity, and neutral pH for a week or more. The growth of the virus is demonstrated by the typical cytopathic effects, which are cell rounding up, detachment from the monolayer, lysis, and release of virus particles into the medium. This is observed under a microscope. Here's a picture showing a monolayer affected by the adenovirus, that is the CPE. If you are keen, you can notice that the cells have rounded up. Here is another picture showing an incubator with cell culture flasks. This cell culture technique is then followed by immunoassays or polymerase chain reaction tests, also known as PCR tests, to confirm the identity of the isolated virus. The importance of the cell culture technique has been evidenced by the development of polio vaccine, the measles, mumps, rubella vaccine, the study of TB and HIV, and in the study of human chromosome number and mitosis, whereby the HeLa cell line was quite useful. Here are my references, and thank you for your attention.